Hey folks, so as you know, I told you I had hand surgery on Friday and I still can't use it, and I'm going to take one more day just to get enough use back so I can drive comfortably. So to keep us on track, I wanted to just do a couple of video lectures that cover the parts of the brain that I needed to either add more detail to or that I haven't covered at all yet. And so I'm going to start with the occipital lobe, and we'll move sort of around the brain and end up at the frontal lobe. So I'm going to start with the occipital lobe, and I just have a few things that I want to add or reiterate to what the group presented us the other day. And so I wanted to remind people that for each eye, we've got two segments of optic nerve, and for each eye, the left part of the eye is a separate bundle of neurons that go back from the right section of neurons. And so for my left eye, the left side of my left eye and I can't move my arm very well, but the left side of my right eye go back together, and this information over here crosses over into my right hemisphere. And likewise, if you flip around and do the right side of each eye, that information goes back to the left hemisphere. And we'll talk more about that when we get to the section on sensation. But for now, just know that anything that's in my left visual field, so over here, is going to go into my right hemisphere, and then it's going to cross back over and end up in my, did I say that right? So if it's over in my left visual field, it's going to end up in my right hemisphere, and information that's over in my right visual field will cross over and end up in my left hemisphere. So it's not information from each eye, it's information from our visual field. And I can clarify that in class if that's not clear. So for each bundle of neurons, we've got information that has to do, where did I put my coconut oil? Information that has to do with shape, information that has to do with movement, and information that has to do with color. And each of those pieces of information goes back on a separate track. And the visual cortex has to merge the information from each of those tracks. Then it has to merge the information from each hemisphere into one. Then it has to sort out the visual information to separate out the foreground from the background. So again, if I take this coconut oil and I put it in front of my face like this, you guys don't freak out and panic and think, oh my gosh, she's got a jar embedded in her face. Your visual cortices interpret this as being in front of my face, because when it sits here, you can see the whole jar, but you can only see part of my face. And so your brain can organize that based on experience and sort it out. And then information has to go around to the temporal lobes to get categorized. And then at the same time, information is shared up into the parietal lobe to get the orientation of things. So you would recognize that this is upside down and backwards actually to you. And then and your parietal lobe would help put things together in space. So the visual cortex actually takes in essentially digital information. Neurons are either turned off or turned on, and they're in a pattern, and the visual cortex has to piece all those things together, merge them, separate out foreground from background, and then ship them around. So by the time we have a conscious awareness of what we're looking at, that information has already gone around to the temporal lobes and up into the parietal lobes. So the visual cortex also plays a role in reading and our understanding of writing and that kind of thing. And our ability to make sense of those depends on our knowledge of language, which our temporal lobe contributes to with Wernicke's area, and that orientation of letters is contributed by the parietal lobe areas in the parietal lobe that contribute to our vision. Okay, so that's occipital lobes. I think the visual pathways occipital lobe group gave you a good mnemonic device that people have eyes on the back of their heads, especially mothers, since today's Mother's Day. Okay, so that's the occipital lobe, visual cortices. If we come around to the temporal lobes, there are a couple of different areas of temporal lobes that you should be aware of. The primary cortex in the temporal lobes is the auditory cortex, and that does for sound what the visual cortex does for vision. So with sound, if you've ever looked at a sonogram, I guess it's a gram or a graph, sound is separated out into amplitude, and that corresponds to volume, so how loud something is. Frequency, which has to do with how many waves you get in a particular unit of measure, and that corresponds to pitch. So high frequency means that there's lots of little waves, and we hear that as high pitch. And long wavelengths, so there's very few in a particular area, unit of measurement, we perceive as being very low, in like deep low. And then the other has to do with the smoothness of the curve, and that corresponds to what the musicians call timbre, T-I-M-B-R-E, timbre. And that has to do with, like, if you're listening to somebody on The Voice or whatever, if they've got like a gritty voice, that would have to do with the smoothness. So if it's clear or gravelly, sort of. So the primary cortex in the temporal lobes is the auditory cortex. But don't forget that there's Wernicke's area there. There's also another part of the temporal lobe that contributes to vision, which you heard in that presentation. And that part of each temporal lobe contributes to categorizing visual information. And there's kind of a debate, but currently people are, well, I'll just leave it that it's a debate. So it categorizes visual information, and one type of visual information are faces. So what our face looks like. And there's, if that area is damaged, whether it's from a traumatic brain injury or if there's some developmental glitch, people can end up with a symptom of not being able to recognize faces. And they call that disorder prosopagnosia. So P-R-O-S-O, P-A-G-N-O-S-I-A, prosopagnosia. And again, that's an inability to recognize faces. And you can have that on a spectrum of really severe and relatively mild. And I have a story that I can tell about my own life and this disorder. So I think a lot of you know who Jane Goodall is. She's one of my heroes, and she's one who has worked in Africa studying wild chimpanzees since the 1960s. And so in my field working with chimpanzees, I've read all of her work, and she was, I've met her a number of times, and she's just a genuinely nice person. And when I worked at the chimp lab up at Central, I actually was in a position of authority, and so I got to spend time with her. And after the second or third time that I'd met her, she started, when she would show up the next time, she would act like she knew who I was. And I was just, it's like, oh my gosh, my hero knows who I am. I must be somebody. And so I was pretty darn proud of myself. And then I went down and worked on my PhD, and my major professors were also friends of hers. And so she came down there, and sure enough, we had a dinner party, and she showed up, and she approached me and acted like she knew who I was, and I couldn't believe it, and oh my gosh, Jane Goodall knows who I am. And so I just thought I was hot stuff. And so years later, I apply for this job at YVC, I get the job, and meanwhile, people had been bugging Jane about writing her autobiography. So she finally capitulated and wrote one. And so my mom got it for me for Christmas, and I was all excited I was going to read it. So I started reading it, and I get to the section of the book where she's talking about her early years in England, and how in her small town she was just humiliated, or felt humiliated, a lot because she would meet somebody and then maybe the next day on the street she would bump into them and she wouldn't recognize them. And she felt so awful. And here I am thinking, oh, but she knows me. So I'm still thinking I'm hot stuff. And so she is like the most genuine person I've ever met. She's just super nice and kind-hearted. So anyway, she said at the time that she was determined not to make those kind of errors. And so she began treating everyone as if she had met them before. So after I cried myself to sleep, I finished the book and sort of got over it and got myself put in my place. Anyway, it turned out that she had prosopagnosia. So she didn't know me, in case you didn't figure that one out. Anyway, so that's prosopagnosia. So a couple other things with the temporal lobe. When we talk about categorizing, it's getting categorized into things like pen-like object, or cell phone-like object, or paper-like object, or face-like object. And then face-like object becomes, somebody that you actually know. And so we actually really can't talk about what we see until that visual information that I talked about in the occipital lobe makes it around into the temporal lobe. And information from the parietal lobe also has to get factored into that for us to talk about it. So the temporal lobe, if you get temporal lobe damage, you can end up with an inability to process visual information. So you can be object blind or face blind, even though your eyes are fine, your retina is fine, your occipital lobe is just fine. The information can make it around, but you still have trouble seeing. So vision is a very complex thing. We take it for granted that we open our eyes and the world makes sense, but that really is, we do take that for granted because our vision can be interrupted at any spot along the pathway of that information coming in and getting processed. Okay. And then don't forget that the Wernicke's area is in the left temporal lobe. And just as an FYI, I'm not going to test you on it, but the part of the temporal lobe that's kind of the visual pathway is called the fusiform gyrus. And it actually runs from the visual cortex, so from the occipital lobe all the way across the temporal lobe toward the frontal lobe. And so that visual contribution of the temporal lobe is in that fusiform gyrus. Okay. So then, let's see. I think I'm going to stop there, and then I will start a second sort of 10-ish, 15-minute lecture on the parietal lobe and the frontal lobe. And if I remember anything that I should have included here, I'll add it to that other one. But I'll go ahead and stop here so that you can sort of have two shorter sittings. Okay, I will see you in a minute.